Good evening, everyone. I welcome you all to the week 7 live problem solving session on NPTEL Bioinformatics Algorithms and Applications course. This is part of the lecture on Monday. So, due to unfortunate circumstances, I wanted to shift it to on 9th March 2025. So, in the last class, we have had introduction to protein structure and also protein structure prediction. So, in this, I'll be starting about computational protein structure analysis. It is a broad field that involves various techniques and tools for studying the structure, function and dynamics of protein. So here I have listed down some of the features of computational protein structure analysis, such as structure prediction, structure alignment, molecular docking and molecular dynamics simulation. One of the primary features of the computational protein structure analysis is predicting the 3D structure of a protein from its amino acid sequence. This involves using algorithms and computational methods to predict the most energetically favorable conformation of a protein. In this regard, in the last class we have seen the introduction to AlphaFold, how a structure has been predicted using the from AlphaFold using the sequence. Then we have structural alignment, which is a process of comparing 3D structures of two or more proteins to identify similarities and differences. It is useful for understanding the evolutionary relationships, identifying the functionally important regions and designing new protein structures. Third one is molecular docking, which is a technique used to predict the binding mode of a ligand to a protein. Ligand can be any of the small molecular drugs that are intended for human application. So molecular docking involves generating different conformation of the ligand and protein and calculating the energy of their interaction. Finally, we have molecular dynamics simulation, which is used to study the motion and behavior of the proteins at the atomic level over the time. It involves solving Newton's equations of motion for all atoms in the protein, which allows researchers to study protein dynamics, conformational changes and protein ligand interactions. So here I have a question which states that which is one of the primary features for computational protein structure analysis, whether calculating amino acid combination is a primary feature, protein structure prediction, or BLAST, that is basic local alignment search tool, or none of the above. Here, amino acid composition and BLAST are based on the alignments and primary structure. So these mostly are performed using the sequences. So protein structure prediction is the primary feature in computational protein structure analysis. In the last class, we also had introduction to PDB param, like a demo. So here the PDB param server provides the comprehensive set of structure-based features derived from protein three-dimensional structures. These features are grouped into four main categories: physiochemical properties, inter-residue interactions, secondary structure propensities, and identification of binding sites in the protein complexes. First class is physiochemical properties. These properties describe various characteristics of protein structures related to their chemical and physical properties. This includes parameters such as center of mass, which defines the overall mass distribution of the protein; radius of gyration, which describes the compactness of the protein structure; surrounding hydrophobicity, which measures the hydrophobic behavior of amino acid residues in the protein environment; hydrophobic accessible area, which calculates the solvent accessible surface area of hydrophobic residues on protein surface; and buriedness, which evaluates the degree to which a residue is buried within the protein structure, and non-covalent interactions. Here, under this category of non-covalent interactions, we have various interactions like hydrophobic hydrogen bonds, ionic aromatic cation pi disulfide present in the protein. Next, we have interresidue interactions. These parameters focus on understanding the interactions between the amino acid residues within a protein structure. So, the key features include short, medium, and long range interactions, which analyze the interactions within the different spatial ranges. We have contact order and long range order, which reflects the importance of local and non-local contacts to the native structure of the protein, and long range order reflects the importance of long range contacts in the protein structure. Then we have total contact distance. This is a parameter derived from the product of contact order and long range order. Then we have multiple contact index, which identifies the residues with multiple contacts in 2 and 3 state proteins. Along with that, third category is secondary structure propensities. Propensity is similar synonym used for the term probability. So these parameters assess the likelihood of amino acid structures to adopt to specific secondary structures such as alpha helices, beta strands, and turns. So features include propensity of a residue to be in alpha helix, beta strands, and coil, frequency of a residue to occur in beta bends and turns, normalized frequency of a residue to be in helix, and the propensity of a residue to form multiple contact index. Under the fourth category, that is identification of binding sites, this helps in locating residues involved in protein DNA, or protein RNA, like oral protein nucleic acid, protein ligand interactions based on the distance criteria between atoms. It provides information on the binding sites within the protein. So this PDB param server offers valuable resources for analyzing protein structures and understanding their functional implications. By providing access to a wide range of structure-based parameters, it facilitates in-depth investigations into the structure-function relationships of proteins. So I have mentioned identification of binding sites. PDB param is a distance-based method. Other than that, we have ASA-based methods and energy-based methods. Recently, we have ML-based methods as well. So overall, the binding sites for a given protein complex is predicted either by distance-based method, ASA-based methods, energy-based methods, or using any of these ML-based methods. So here I have given the steps like how to compute a center of mass of a protein. So given the PDB ID is 2TRX and from chain A. So PDB param is a structure based method, so it requires either a structure input or a PDB ID as an input. So this is under physiochemical properties. Let's go and see the demo of this. I type PDB param. So this is the page. So as I mentioned, it is available under physiochemical properties. So I go first, click on compute, then enter the PDB ID. As I mentioned, the PDB ID is 2TRX, chain is A. So since I want to calculate center of mass, it is under physiochemical properties, and then select center of mass, and it is we have also option to print all the results. Submit. Yes, please.
Excuse me, sir. I would like to know if PDB param gives information about docked molecules also. Like if I have a docked molecule, already I have docked between protein protein by protein protein interactions. then PDB param will give information about that.
Yeah, actually, we have structure upload module for PDB param as well. So you can upload your structure and you can get this binding sites, identification of binding sites you can perform.
Okay. We can upload the structure, any structure we can upload.
Yeah, it's in a PDB format, acceptable PDB formats. PDB param
PDB
will.
format. Okay. 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 Thank you, sir.
So here the results page shows the input PDB ID and the chain, followed by molecular weight in kilodaltons, followed by center of mass. So the x and z, x, y, and z coordinates. So here the screen I'm showing the formula of the center of mass that runs in the back end of the PDB param code. So it is summation of mi(xi) divided by summation of mi. So where m is the atomic mass and xi is the Cartesian coordinate. So this is for x coordinate. Similarly, y center of mass and z center of mass will be calculated. So here the I value runs from 1 to n, where is total number of atoms. Yeah, so these are the steps that we have seen in the server. So using PDB param, yeah, try to report the following number of interactions for a PDB ID to TRX chain A. So the interresidue interactions are also part of physicochemical properties. So I have mentioned we have disulfide, ionic, hydrophobic, and aromatic. Ionic, hydrophobic, and aromatic. So under disulfide bridges, we have one disulfide bridge that is formed between residue, cysteine residue at the 32 position and cysteine at 35. The distance between, so the bond length or distance between this disulfide bridge is approximately 2.1 angstroms. Then we have set of ionic interactions. So we have 21 ionic interactions followed by hydrophobic interactions and aromatic interactions. So this is how a PDB param provides interactions. So these are the number of interactions. Like we have one disulfide bridge in chain A of 2TRX, and we have 21 ionic interactions, and we have 16 hydrophobic interactions and 4 aromatic interactions. So here I have a question. So which of the following category does not belong to PDB param server functions? So whether physiochemical properties, molecular dynamic simulations, interresidue interactions, or identification of binding sites. Yeah, so molecular dynamics is not a feature provided by the PDB param server. Yeah, then we'll start with the protein structure alignment. Sequence alignment we have studied. So we have studied pairwise sequence alignment. Under pairwise sequence alignment, we have studied dot matrix and dynamic programming methods such as Needleman Wunsch and Smith Waterman algorithms for global and local alignments, and then we have studied multiple sequence alignment as well. So protein structure superimposition is a process of aligning two or more protein structures to compare their similarities and differences. It is performed by several steps. First is least square fitting. So in least square fitting, imagine you have two protein structures and you wanted to see how similar they are. Least square fitting aligns these structures by finding the best possible match for their C alpha atoms. It does this by minimizing the overall distance between the corresponding C alpha atoms in both structures. Essentially, least square fitting tries to make structures overlap as much as possible with least amount of bending or twisting. While it is quick and straightforward, it may miss some finer details in the structure. So we have dynamic programming, which is a method which is more flexible and handle complex structural differences. It uses a matrix and compares the different parts of the structure, gradually adjusting them to find the best overall alignment. It is like solving a puzzle where each piece of the structure is moved and rotated until the best fit is achieved. However, this approach can be slower and requires more computational power. Then we have iterative methods which refine the alignment of protein structures through iterative process. It starts with an initial alignment and then repeatedly adjusts it based on the local structural similarities. It is like gradually fine tuning the alignment until it fits perfectly. This approach is more accurate than least square fitting and can handle more complex structural differences. So other than this, we have multiscale methods and geometric uh, hashing methods. So multi-scale methods take an hierarchical approach to the alignment. They compare protein structures at different levels of detail from large-scale features to small-scale details. This allows them to handle large structural differences between the proteins while still capturing final similarities. It's like zooming in and out on a picture to find matching patterns at different levels of magnification. As I mentioned, geometric hashing focuses on identifying similar structure motifs between proteins. It represents the structures in a geometric way and looks for a shade motifs that can be used to align them. It is like finding a common shapes or patterns in two different pictures and using them to align the pictures accurately. So this method is particularly useful for handling large structural differences between the proteins. So here I'll discuss some of these methods. So as I mentioned this, the least square fitting method primarily aims to align the C alpha atoms in protein structure. So that is backbone C alpha atom. So we have distance matrix alignment method, shortly known as DALI, which is used for comparing and superimposing protein structures. It compares structures by aligning their secondary structure elements and assessing overall topology similarity. Steps in DALI include uh, first one is distance matrix calculation, then identification of secondary structure elements, then alignment optimization and similarity score calculation. So DALI computes a distance matrix between the pairs of C alpha atoms in protein structures. Then it identifies the SSEs, that is secondary structure elements in each of the structures, and aligns them based on their spatial arrangement. And then alignment is, this spatial arrangement is optimized by minimizing the RMST between these aligned SSEs, that is, it tries to minimize. Finally, it calculates a similarity score considering the number of aligned residues, RMST, penalty for gaps, and insertions. This method DALI assigns a z-score to each pairwise comparison, indicating the structural similarity significance based on the scores from a database of known protein structures. This is widely used in structural biology for comparing and classifying protein structures. It is specifically used for finding proteins with similar folds but low sequence similarity. And DALI is also integrated into various software packages, like we have DALI server, DALI Lite program for easy access and usage. And it also this method also demonstrated the reliability and accuracy in protein structure comparison tasks, making it a valuable tool in structural biology research. So here I want to recap and summarize the X-ray, NMR, and cryo methods. So as we all know, many of the structures that are deposited in the current protein database PDB is from X-ray, which is a powerful method for determining the three-dimensional structure of proteins at atomic resolution. It relies on the diffraction pattern produced when X-rays interacts with the ordered array of atoms in protein crystal. X-ray crystallography provides high-resolution structures capturing detailed atomic positions and bonding interactions. However, this method requires the formation of well-ordered protein crystals, which can be challenging for certain proteins. Additionally, the process of data collection and structure determination can be time consuming and resource intensive. Then we have NMR. So in this method, by analyzing the behavior of atomic nuclei in a magnetic field, this method provides the information on distance between the atoms, dihedral angles, and interactions within the protein. NMR can be applied to proteins in their native state, allowing the study of dynamic processes such as folding and binding. However, NMR is limited by the size of the protein, as signal overlap becomes problematic for larger molecules. Additionally, the requirement of stable and soluble protein samples can pose a challenge in the application of NMR spectroscopy. The recent and now currently wide used is cryo-EM. Currently, if you see the recent deposits on PDB, we observe many of them are from cryo-EM, as it is emerged as a versatile technique for visualizing the structures of macromolecular complexes including proteins. So in this method, by rapidly freezing samples in vitreous ice, cryo-EM preserves their native state and allows imaging under near physiological conditions. This cryo-EM can achieve medium to high resolution structures, making it suitable for studying large protein complexes and flexible structures. However, cryo-EM requires specialized equipment and expertise for sample preparation and data analysis, and achieving high resolution structures may be challenging for certain samples. Yeah. Experimental methods for protein structure determination such as X-ray, NMR, and cryo-EM are incredibly powerful techniques. However, they also have certain limitations. Experimental methods can be expensive and time consuming, and obtaining high resolution techniques through X-ray, NMR requires specialized equipment, skilled personnel, and significant resources. Moreover, the process of protein expression, purification, crystallization, and data collection can take months or even years. Experimental methods often require substantial amount of purified protein sample. This can be challenging, especially for proteins that are difficult to express or purify in large quantities. And also, experimental methods may not always provide complete structural information, especially for large and flexible proteins. Regions of proteins that are disordered or dynamic may be poorly resolved or entirely missing from the structure. And we also have protein engineering limitations. In some cases, modifying the protein sequence or structure to facilitate experimental studies may alter its native conformation or functional properties, affecting the biological relevance of the obtained structure. To address these drawbacks, computational methods for protein structure prediction such as homology modeling, ab initio modeling, and molecular dynamics simulation are often employed. These computational approaches can complement the experimental techniques by providing insights into the protein structure, dynamics, and interactions, especially in cases where experimental methods face challenges or limitations. In comparison to computational structure prediction methods, such experimental methods can provide high resolution structures but are often time consuming. So, computational methods use algorithms and modeling techniques to predict the protein structures based on amino acid sequences and known structural principles. Computational methods are faster and more accessible but may have limitations in accuracy, particularly for large or complex proteins. Overall, each experimental method has a strength and limitation, and the choice of method depends upon factors such as protein size, stability, and desired level of detail in the structure. Computational methods complement experimental approaches by providing rapid predictions and insights into the Structural dynamics. So protein 3D structure prediction is a computational method that aims to predict the 3D conformation of a protein from its amino acid sequence. There are several computational methods available for protein 3D structure prediction, including homology modeling, also known as comparative modeling, is the most widely used method for protein structure prediction. It involves predicting the 3D structure of a protein based on its homology to a known protein structure. Homology means similarity. This method relies on the assumption that protein structures are more conserved than the sequences, and therefore two proteins with similar sequences are likely to have similar structures. Homology modeling method requires a template with high sequence similarity to the target protein and can generate accurate models when the template is of high quality. Then we have ab initio method, also known as a de novo methods, predict the protein structure without using a template structure. These methods use a physics-based approach to predict the conformation of a protein by optimizing its energy landscape. This ab initio methods is computationally intensive and requires a large amount of computing resources, but it can be applied to proteins with no homology to any known structure. Ab initio methods can generate models with low accuracy, but their accuracy can be improved by combining them with experimental data. Thirdly, we have threading. Threading is a hybrid method that combines both homology modeling and ab initio method. So the method uses a threading algorithm to search a library of protein structures for the best match to the target protein sequence. The algorithm then generates a model by aligning the template structure to the target sequence and refining it using an ab initio approach. Threading can generate accurate models when a suitable template structure is available in the library. Finally, we have machine learning or deep learning methods. So these machine learning or deep learning methods, ML based methods, use large amount of protein structure data to train a model that can predict the 3D structure of a protein from its amino acid sequence. Machine learning methods can generate accurate models with high speed, but they require large amounts of high quality training data. Each of these methods has its own advantages and limitations and is best suited for different types of proteins and structural features. The choice of method depends on the specific requirements and constraints of the analysis. So these are the basic four classes that are available. So before moving, I would like to explain about homology and steps in homology modeling. So the first step in the homology modeling is identifying and selecting an appropriate template from protein data bank. To identify appropriate template, pairwise sequence alignment methods such as BLAS can be used to identify proteins with high sequence homology to the query protein in the PDB. More sensitive methods involve performing multiple sequence alignment, or automated iterative methods such as SciBLAS can also be included. If the homology between the protein of interest and the potential template is low, alternative methods such as profile alignments or HMM, hidden Markov models, are used to reduce the gaps in alignment. Next is the alignment of query sequence with the template. This is an important step in the process. It is essential to achieve a high quality sequence alignment as it directly impacts the accuracy of the resulting model. A homology model can be only be good as the sequence alignment. So multiple sequence alignment is preferred as it contains evolutionary information that is important for structure and function. Effective multiple sequence alignments like T coffee, cluster omega, cluster W omega are recommended for use. Sequence alignment is followed by the 3D model building of the target proteins. There are different methods used for generating 3D models of a protein based on its template. These methods are classified into four categories: rigid body assembly, segment matching, spatial restraint, and artificial evolution. methods. In rigid body assembly, the protein structure is broken down into basic conserved core regions, loops, and side chain. The rigid body parts are picked up from the template protein structures and brought together using tools like 3D Jigsaw, Builder, and Swiss Modeler. Next, segment matching involves using a cluster of atomic positions from the template structures as leading position. These atomic positions are used to select segments from known structures in the database based on the sequence identity, geometry, and energy. These segments are aligned with the template structures to create a model of the target protein. This can be done using by SecMod or NCAD. Finally, spatial restraint method, which builds the protein structures by applying some spatial restraints to guide the building of a model that closely matches the template structure. Then we have loop modeling. There are often gaps or insertions that can occur in sequence alignments when modeling protein structures. These gaps are called loops or structurally less conserved regions during evolution. Loop modeling is an important step in protein structure prediction because loop plays a crucial role in determining the functioning of a protein. However, predicting the structure is a complex process. There are two main methods for loop prediction, that is database search method or conformational search method. The database search method involves the comparing the target protein sequences with all known protein structures to identify the segments that can be used to model the loop. Then we have conformational search approach, which involves optimizing a scoring function to find the best possible loop conformation. So this conformational search method generates many random loops and searches for the one that fits the target protein structure. This approach is also called as ab initio because it does not rely on pre-existing structural information. Then we have sidechain modeling. Sidechain modeling is a process of predicting the conformation of the side chain of amino acids in a protein structure. This process is crucial in evaluating protein ligand and protein-protein interactions. This is usually done by placing the side chain onto the backbone coordinates that are derived from a protein structure or ab initio modeling simulations. Searching every possible conformation of a side chain is computationally time consuming and not effective. So most sidechain prediction programs use preferred conformations called rotamers. These rotamers are stored in the rotamer library, which is a collection of preferred sidechain conformations ranked by their frequency of occurrence. Different energy functions and search strategies are used to select the most appropriate rotamer for each amino acid sidechain based on the preferred protein sequence and the given backbone coordinates. Next step is to optimize. Now that we have constructed the backbone and we have constructed the sidechain, next is to optimize the model. Optimization of a model refers to the process of refining the initial model to improve its accuracy and reliability. This optimization process involves adjusting the positions of atoms in the model to reduce any clashes or steric hindrances between the atoms. Energy minimization process is one of the methods of model optimization. It calculates the potential energy of the model and adjusts the positions of atoms to minimize this energy. Excessive energy minimization can often move residues away from their correct positions, so only limited energy minimization is recommended. In addition to energy minimization, other optimization techniques such as MD simulation or Monte Carlo simulations can also be used. After the 3D model of a query protein has been built, it is essential to validate and evaluate its quality to ensure that it's biologically relevant and can be used for further studies. This involves the comparing the model to the real experimental 3D structures and using various evaluation methods to assess the model's stereochemistry, physical parameters, knowledge-based parameters, statistical mechanics, and other criteria. So some commonly used programs for evaluating protein structures are WhatIf, ProCheck, and ProSA. So these programs compare the properties of the homology model to those found in the experimental structures in PDB. The quality of homology model largely depends upon the accuracy of sequence alignment and part of the protein where template is not similar to the query protein. So before moving on to ab initio method, let's see. So Swiss model is an homology based. Let's go in. So Swiss model is a fully automated protein structure homology modeling server. It is accessible via Expressive web server. The purpose of this server is to make protein modeling accessible to all life science researchers. So here to start, click on start modeling from the home page. Click on start modeling. So here it asks for a uniprot uh, sequence or any protein sequence as a input. So okay, let me go to uniprot and take any sequence. Let me select a shorter fragment only. Trial 1. So the first is search for templates to obtain a very good sequence alignment. Then click on search for templates. It will run for some time to give the list of templates. Okay, yeah. Meanwhile this runs, I'll move forward the slides. So next type is the ab initio method. Ab initio protein structure prediction methods are computational approaches that aim to predict the 3D structure of a protein solely based on its amino acid sequence without relying on any prior knowledge of its structure and homologous sequences. Ab initio methods are particularly useful for predicting protein structures that have no homologous structures in PDB. The basic idea behind the ab initio protein structure prediction is to generate a large number of 3D conformation of a protein, then to search for the conformation with the lowest energy, which is assumed to be the native structure. The energy of these conformation is calculated using physics-based energy functions that takes into account the electrostatic, van der Waals, and other physical interactions between the atoms. Steps involved. The first step in ab initio protein structure prediction is to generate a large number of short protein fragments, typically 3 to 9 residues in length, that represents the possible building blocks of protein structure. Next step is to assemble these fragments into 3D conformation of a protein. This is done by performing a Monte Carlo search through the conformational space, which involves randomly selecting and rotating fragments and evaluating the energy of the resulting conformation. So this fragment assembly and sampling, this process is repeated thousands or millions of times to generate a large number of possible conformations. Once a large number of conformations has been generated, their energies are evaluated using a physics-based energy function that takes into account the interaction between the atoms. The lowest energy conformation is assumed to be the native state structure. The final step is to refine the predictor structure using energy minimization or molecular dynamics simulation to optimize and remove any steric clashes. So these ab initio methods can be computationally intensive and requires powerful computational resources. However, they can be very useful for the structure of a proteins that are difficult in experimental studies and doesn't have any homology. So example include Rosetta, ITASR, and Quark. So in last class we also started discussing with the AlphaFold, which is an AI program developed by DeepMind that predicts the three-dimensional 3D structure of a protein from its amino acid sequence. So this Rosetta and DeepMind's AI have been recently awarded Nobel Prize for their work in protein structure prediction. So this program uses artificial intelligence based AlphaFold program uses combination of deep neural networks and genetic algorithms to model protein folding. So a process that is critical for understanding protein function and designing. So we have some steps. The first step is to pre-processing. AlphaFold to compares the input amino acid sequences with sequences from database to identify similar ones, detecting correlated mutations and patterns. It also identifies proteins with similar structures to derive the structure information. Then it has overformer module, which is a transformer type of module. So the first we have MSA transformer. So one transformer processes MSA data, extracting information about sequence variations and correlations. Yes, please.
Sir, deep mind that you are talking about now,
Yeah,
and the previous is PDB param that you spoke before. Like deep mind is more advanced than PDB param. Is it like that?
yeah. Okay. Here, PDB param is a structural analysis tool. AlphaFold is a structure prediction tool. So we can, if
Okay.
with using this AlphaFold or Swiss modeler or ab initio methods, we can generate the structure if we don't have any experimental structure. PDB
Okay.
param is like extract features from the structures, whether it's an experimental structure or a predictor structure.
Okay. Here we can extract that structure itself.
Yeah, you just input the sequence, just like
Okay. Fast as just like you did before.
yeah. I'll also show demo of AlphaFold once I go in sequential ab initio and I'll show that.
Okay, sir. Thank you, sir.
Yeah. Then we have this pair representation transformer, which focuses on pairwise interactions between the amino acids and the protein structure. Then we have this final model we call residue gas model. So this structural model treats the protein as a collection of triangles in 3D space, iteratively transforming them into a final protein structure. And we have invariant point attention, that is IPA, which incorporate three-dimensional structural information using a novel attention mechanism, enhancing the model's learning and prediction ability. Okay, okay. Before moving on to stability, yeah. So here initially we have given a input of sequence and we search for a template. So these are the templates that are high have high alignment or high score. So if you see identity, the template one, which is from amyloid beta precursor of human, has 100% identity, and this structure is already from AlphaFold. So AlphaFold has a database as well, which has already pre-computed structures. Next, we have again amyloid beta A4 protein, which is X-ray. It has structural resolution is 2.7 angstroms. So now we can select the template and ask it to build a model. So for our given input sequence, this is the structure, yeah, that has been predicted by the homology modeling. Now whether we should know whether the structure is good or no. So here we have structure assessment option. The first assessment is to find the deviations from the Ramachandran plot. Then we have different scores, that is small probability score, clash between the side chain score and Ramachandran favored. So here we have seen there are some outliers in the structure which doesn't follow the Ramachandran's principle of psi and phi angles. And we have rotamer outliers also. This rotamer outliers, C beta deviations, bad bonds and bad angles are based on the clashes. So lesser the number of outliers and lesser the number of clashes, the more accurate or good is the quality of the predictor structure. So this is one such method. Then now we have ITASR, which is ab initio based method. So ab initio methods are preferred if our given protein sequence doesn't have homologous sequences. So because as I mentioned, ab initio methods take time, I'm using their example, one of their given example in their website to demonstrate now. Yeah. So first the result page shows what is the sequence submitted. So this is the sequence submitted. Then it assigns the predicted secondary structure for our given sequence, input sequence. Secondary structure annotations has been done along with the confidence score. So in the secondary structure annotations, H is for helix, S is for beta strand, and C is for coil. Then again it does predicted solvent accessibility. So the predicted solvent accessibility values ranges from 0 to 9. 0 stands for buried residue and 9 stands for highly exposed residue. Then we have predicted normalized B factor as well. Then it uses threading templates. So in threading method, as we studied, it's a combination of ab initio as well as the homology method. So it identifies 10 uh, PDB IDs, that is existing methods, and it tries to construct a multiple sequence alignment following the secondary structure comparison. Finally, based on this, top 5 models has been given as an output. Okay. Yeah, in these empty spaces actually structure should be shown, I think. Yeah, there is some issue in there. So these are the final models, so we can download these models and use them and view them in PyMol. Okay, now that I'll move to AlphaFold server as well. So AlphaFold has a server which has been recently generated. I think in the last class we have done this AlphaFold. I'll just take that. So given the input sequence, AlphaFold predicts the structure. Sorry. So AlphaFold can predict the structure of a protein, DNA, RNA, ligand, or ion, or it can also predict the structure of complexes, that is protein complexes, protein nucleic acid complexes, and protein ligand complexes. So here I have made a sequence. Okay, let me more reduce the sequence for faster run. So I have selected entity type protein, number of copies. So these copies means number of chains. So now a structure will be generated with one chain. I submitted the job. Okay, let this result run and proceed with protein stability. So here I want to introduce a concept called protein stability, which refers to the ability of a protein to maintain its native conformation or structure under the range of environmental conditions, such as changes in temperature, changes in pH, ionic strength, and pressure. The stability of a protein is important for its function as well as for the design of protein-based drugs or therapeutics. Protein stability is determined by the complex interaction of various factors including protein structure, solvent environment, covalent modifications, chaperones and other proteins, ligand and substrate. Overall, the stability of a protein is a complex and dynamic property that is crucial for its function and biological activity. Understanding the factors that contribute to the protein stability can help in design of more stable proteins for the therapeutic application. So one of my current research motive is understanding the factors contributing to the stability of membrane proteins and also assessing the impact of these factors on the protein stability. So when we come to the contributions of protein stability, we have hydrophobic effect. Hydrophobic effect plays a crucial role in stabilizing the native state of a protein. When non-polar amino acid residues are buried in the hydrophobic core of the protein, they minimize their exposure to the surrounding aqueous environment. This minimization of hydrophobic surface area reduces the unfavorable interactions between non-polar residues and water molecules, thereby contributing to the overall protein stability. And van der Waals interaction also have effect. The burial of these hydrophobic effect in protein's core reduces the entropy of surrounding water as they become more ordered around the non-polar groups. This entropy decrease is favorable and contributes to the overall stability of the folded protein. Yeah, then we have this van der Waals interactions and hydrogen bond interactions. So they mainly affect the enthalpy, and the opposing force of the folding is the conformational entropy. So the balance between the forces promoting folding and opposing folding is the net protein stability, and it is a very small value, and this value hardly ranges from 5 to 25 kilocal per mole. Okay, let me move on here. Alpha fold. Yeah, so our alpha fold structure is predicted. So for our given input sequence, this is the given structure. So the color code here tells the quality of the structure. Very high PLDT is a very accurate or good prediction. Low is very bad prediction. So our complete structure has a very good structure. So now let me go back and try to increase the copy number. So here I am increasing the copy number. So now for the given, previously we have used one chain. Now two chains will be formed. Let's go quickly through these questions. So which of the following structure prediction method does not rely on a template structure? So homology modeling. Thank you. It is Ab initio modeling. So ab initio modeling is based on the physics-based energy functions and physicochemical properties of amino acid sequence. It does not rely on the template structure. Which of the following is dominant in unfolded state of a protein? So, so one can consider green is for folding and red is for unfolding. So which of the following is dominant in the unfolded state of a protein? So forces or interactions such as hydrophobic, van der Waals and electrostatic. No, actually hydrophobic collapse is a driving force for folding. So when all these hydrophobic residues in unfolded state are exposed to water, so because of this non-polar nature and polar water, water movement is restricted. So when these hydrophobic residues are moved away from the water into core of a protein, the water around this protein moves very freely. So their entropy is increased. So entropy is the dominant in the unfolded state. So the protein's entropy is lost and the water entropy is gained. So this is the balance between the entropy as a driving force for protein stability. So which of the following interaction is modeled using LJ potential or Lennard-Jones potential during MD simulation? So LJ potential is mainly about van der Waals interaction. So it's a distance based with R is used. So which of the following methods are used for measuring folding free energy? This is just an intro to our next class. Protein stability and protein stability methods and computational methods for predicting protein stability will be discussed in the next class. All of these methods, circular dichroism method, differential scanning colorimetry and fluorescence spectroscopy are used. So here I have a true or false question. Rosetta is a web server that predicts the protein three-dimensional conformation using ab initio method. It's true or false. Yeah, true. It's an ab initio based method. So which of the following is used for validating protein model structures? Whether ProCheck is used, check PDB is used, Swiss model is used, or all of these above. Yeah, ProCheck. Swiss model also again refers to the ProCheck. To it's a what we call it's an extension of ProCheck that Swiss model has. So ProCheck is the one that is used for validating protein model structures. Swiss model also uses ProCheck. So here I have a someone brainstorming question, which is like you have a protein sequence and you want to quickly predict its structure. After performing blast searches, you identify that most closely related proteins with a known structure are having only 50% sequence identity to a protein with a non-significant expect value, that is z value. So which of these options is best? So ab initio method, because we don't have any proper identical protein structure that is available, so ab initio method is good. So here I have given center of mass question for alanine. So I have given x, y and z coordinates. So can you calculate the center of mass? The formula for x coordinate has been given.
Excuse me, sir. The question cannot be seen properly.
This one, it's fine now.
Sir, it's minimi. I mean, it's very not zoomed.
No, like in my other screen, I can find this question good.
Okay. But subtitles are more clearer. I don't know why. Maybe my screen. Okay, sir.
So here I have given a PDB format, that is atom x and z coordinates for a atoms in residue alanine. So I want uh, what is the center of mass. So center of mass for x, center of mass for y, center of mass for z. So as I mentioned, center of mass is summation of mass of an atom into coordinate of an atom divided by the mass of the atom. So here I generate the formula. So value atomic weight of N is 14. 14 into x coordinate 18.6 plus 12 into, which is atomic weight of C. C alpha is again a carbon atom. 12 into 17.15. Again we have C. 12 into x coordinate of C atom, and atomic mass of oxygen into x coordinate of oxygen atom, and again C beta atomic weight into x coordinate, and divided by sum of all the atomic masses. Atomic mass of nitrogen, carbon, C alpha, uh, carbon, C, oxygen, and C beta. So this is how a center of mass for x coordinate has been done, and the y coordinate formula is here. Please go through and solve this solution on yourself. And meanwhile, I'll go to this alpha fold. Yeah, previously we have a single chain structure. Now we have two chains because we have given number of copies has 2. As the number of copies increased, the structure quality decreased. So previously we had a very high PLDT score for our structure. Now the structure quality has been decreased. This is because we don't have a proper aligned dimer or two chain protein for this. Yeah, that's all from me today. If any other questions, I can,
Sir,
yeah.
I have one question, though it is out of the scope of this class.
Yes,
May I
yes,
carry on?
yes, yes.
Sir, like admit analysis, as we have no in bioinformatics.
Hmm.
For that, I have docked a molecule, like I have a docked molecule, and how to find out the admit analysis data from their docked molecule, protein docking molecules.
Yeah, so you have a protein as a small molecule.
No, I have subsections. Like within one docked molecule, there are five subunits.
So for
Sub.
each individual, you can also check Swiss ADMET. Swiss ADMET should work for small molecule.
So each one I have to check, sir.
That's what. First thing is whatever you are docking, you should check. For the protein and all, if you are taking a reference protein,
Yes.
you need not check for those reference protein. If any molecule you are docking, you should check.
Yes, sir. Like after docking, I have five subunits. Okay, sir. like how to check the admit analysis for that? I'm unable to understand.
No, see, admit is for properties are for a compound. Like for example, if you are generating a new drug or a new protein as a drug, if you are giving, so it's for a
Hmm.
compound, not for a complex.
Okay.
ADME properties, we don't measure, like general toxicity is measured for complexes, but other ADMET, absorption, distribution, metabolism, and excretion properties are checked for a compound itself, single compound itself. So you,
Means there is after interactions are over, there is nothing to compare. Means
no,
admit, in
yeah,
terms
that's
of
what,
admit.
that's what that these admin properties, in my knowledge, they are unaffected by these interactions. So either people go through a library of compounds and select the compounds that cross what that interact with their uh, protein of interest or anything that they have good interaction in docking, then they filter these good interaction proteins and check their admit properties. If their admit properties are good, then only they report them. If the admit properties are not good for that compound, they won't report them.
Okay, sir. Means you are saying that admit should be checked before docking.
Yeah, admit can be checked before docking or after docking also admit can be checked, but only reporting of the results will be done if they uh, what pass the admit test.
Okay. Thank you so much, sir.